Hello students, welcome to EPG Pathshala. I am Pooja Arora from Hansraj College, University of Delhi. Today we are going to discuss about a module T-cell development from paper Immunology. So students, let us see what we are going to learn from this module. T-cell development module will help us to understand how T-cell acquire its receptor, TCL receptor, with the help of which it can recognize antigen presented on MSC molecules. We will also learn how T cells are educated about self-MSC molecules, how they attain self-tolerance, and this knowledge will help us to understand the strategies and how those strategies, with the help of those strategies, we can manage acceptance of foreign organ transplants. The knowledge will also help us to understand how TCR expression pattern happens in T cell, which help us to devise strategies to transfer T cell in immune compromised patient such as HIV. So, first of all, a brief introduction to T cell. So, T cell at a glance. Among several B and other cells, T cell constitute major proportion of blood cells. They play central role in adaptive immunity. The distinguished characteristic of T cell is that they can recognize antigen displayed on MSC molecule, same as that of host MSC from which they are isolated. So, T cell demonstrate self-MSC restriction. Mostly, T cells don't activate in response to self-antigens of host from where they are isolated. Hence, they are said to be self-tolerant. T cells have T cell receptor with the help of which they can recognize antigen on self-MSC molecules. Along with TCR, they have CD3 complex which helps in transducing downstream signaling after antigen binding and leads to activation. There are majorly two types of T cell, alpha beta and gamma delta T cell. Most of the T cell have TCR receptor with alpha and beta chain. Those are known as alpha beta T cells. A few percentage are present with gamma and delta chain. Those are known as gamma delta T cell. Gamma delta T cells differ from alpha beta T cells in antigen recognition as they have very limited antigen diversity and they can recognize antigen presented by CD1 molecule. It is MSC-like molecule. While alpha beta T cells have CD4 or CD8 co-receptors which further help them in antigen recognition process. In fact, CD4 displaying T cell, also known as T helper cell, can interact with antigen presented on MSC class 2 molecule. So, CD4 T cell are class 2 MSC restricted, while CD8 T cell, also known as C cytotoxic cells, are class 1 MSC restricted. The gamma delta T cells don't have CD4 as well as CD8 co-receptors. T cell development. T cell development. T cell originate in bone marrow from hematopoietic stem cell by the process of hematopoiesis. Hematopoietic stem cell give rise to myeloid progenitor cell and lymphoid progenitor cell. Lymphoid progenitor cell further differentiate into T cell progenitor cell along with other cells such as B cells, dendritic cell, etc. T cell progenitor cell further move to thymus where they get mature. Stages in T cell development in thymus. Thymocyte interact with stromal cells of thymus such as epithelial cell, dendritic cell, macrophage cell, and their interaction contribute to growth and maturation of thymocytes. The major changes in thymus include arrangement of germ cells, TCR genes, followed by acquirement of functional TCR gene. Means in thymus only T cell acquire their functional T cell receptor complex. T cell also express specific cell surface membrane marker such as CD4 and CD8, and this expression also happen in thymus. In thymus, they get trained by thymic education. This include recognition of MSC molecule on thymic stromal cell, thus becomes self-MSC restricted and non-reactive to self-antigen, thus becoming self-tolerance. This training is popularly known as thymic selection. T cell development in thymus. T cell progenitor cell goes through various double negative stages, double negative 1 to double negative 4 stages. T cell progenitor cell in thymus first undergo proliferation. It passes through double negative 1 and double negative 2 stage. Double negative 2 specialize to form double negative 3 with reduced expression of CD44, increased expression of CD25 and RAG genes. Double negative 3 cells with successfully completed beta chain downregulate CD25 expression, which further differentiates into pre-T cells where beta chain combined with 33 kilodalton pre-alpha chain to form double negative pre-T cells with pre-alpha beta TCR receptor. T cell progenitor cell in thymus, first it has various markers such as c-KIT. c-KIT is a stem cell growth factor marker. CD44, it is addition molecule which is a specialized for homing. T cell progenitor cell doesn't show CD4 or CD8 co-receptor expression, that's why it is double negative, and it doesn't show TCR gene expression. So it is double negative 1 stage. T cell progenitor cell in double negative 1 stage proliferate for some time and which further develop into double negative 2 stage where it starts showing increased expression of CD25, which is a alpha chain of IL-2 receptor. It also shows the expression of c-KIT and CD44, but it is still CD4 and CD8 co-receptor negative. That's why it is also double negative 2. Double negative 2 stage proliferate and differentiate further where it lowers down the expression of c-KIT. Also CD44 expression goes down, but it CD25 expression goes up. At the same time, it start expressing RAG1 and RAG2 genes, the protein of which is required for successful rearrangement of TCR genes, alpha and beta chains. And still the cell is double negative 4 CD4 and CD8 co-receptor. The thymocyte in this stage is known as double negative 3 stage. When double negative 3 cell, it start rearranging beta chain. So successful double negative 3 cell with rearranged beta chain becomes double negative 4 stage where it has rearranged beta chain, but CD25 expression goes down and it does not show the CD4 and CD4 expression, so again it is double negative. There are few cells, double negative 3 cell, which instead of beta chain they start showing expression of gamma chain. The percentage of those cells is very less. Now the cell which is in double negative 4 stage with successful rearranged beta chain further differentiate where it combine with 33 kilodalton pre-alpha chain to form pre-TCR receptor, and the cell with pre-TCR receptor CD3 complex basically combine together and it becomes pre-T cell. pre-T cell with pre-TCR receptor interact with thymic stromal cell such as epithelial cell, dendritic cell, macrophages, etc. It interacts with class 1 MSC or class 2 MSC molecules along with intrathymic ligand, and this interaction leads to the signaling, downstream signaling in pre-T cell, and this signaling results in no further rearrangement of beta chain. It helps in increased expression of CD4 and CD8 co-receptor. It also makes cell permissible for rearrangement of alpha chain genes, and the pre-T cell proliferates and then it also rearrange alpha chain gene, and the cell now has rearranged alpha beta chain CD3 complex, and now it is double positive for CD8 and CD4 co-receptor. The cell in this stage is pro-T cell, or it is also called double positive T cell. Pro-T cell, which is double positive for CD8 and CD4, now interact with thymic stromal cell such as macrophages, dendritic cell and epithelial cell, and this interaction with the thymic stromal cell leads to its selection, and that is also known as thymic education or thymic selection. This selection helps in formation of T cells which are self-MSC restricted and which are self-tolerant. Self-MSC restriction means the T cell can respond to MSC of the host cell in which they are generated. So they are self-MSC restricted. And self-tolerance means the T cell will not get activated in response to self-antigen present in host cell. So they are self-tolerance. So this selection happen in thymus. So pro-T cells, first positive selection means selection for only those T cell which are self-MSC restricted. It happens in cortical region of thymus where it interact with thymic epithelial cell. Now pro-T cell which interact successfully with thymic epithelial cell either via class 2 or class 1 MSC molecule, they will get some signal, protective signal, so they will survive, and those cells which have not received any signal or which have not interacted with thymic epithelial cell will undergo apoptotic death. So this phenomena where only those cells have been selected which interact with epithelial cell, so those cells will survive, and this phenomena is known as positive selection. Negative selection. Negative selection occurs for positively selected thymocytes. Here, thymocytes which interact very strongly with MSC complex alone or with self-peptide MSC complex displayed on the surface of dendritic cell and macrophages of thymus die by apoptosis, while other cells get mature into single positive CD4 or single positive CD8 cell. This phenomena makes sure that any cell which activate with self-antigen presented by MSC molecule or self-MSC molecule very strongly, they die. So it helps to maintain the phenomena of self-tolerance, and this phenomena is known as negative selection. Early evidence for the role of thymus in the selection process of T cells came from chimeric mouse experiments by Zingernagle and his colleague. These researchers implanted thymoectomized and irradiated A into B F1 mice with B-type thymus and reconstituted animals' immune system with an intravenous fusion of F1 bone marrow cells. Here, A into B F1 bone marrow transplant mature within a thymus that express only B haplotype MSC molecules on its stromal cells. Now to confirm whether T cells now recognize or get restricted to MSC haplotype of thymus, in order to find out that these mice were infected with LCM virus and spleen was taken out, and where immature T cells were tested for their ability to kill LCM infected target cells from the strain F1 and from the strain B. It was found that they could lyse the target cell which express the haplotype B, which was the same which was there in the thymus haplotype. But when they were tested again target cells from F1 strain, there was no killing. So it indicates that they can recognize the haplotype which is shown to them in thymus. So they become that haplotype restricted. So thymus play important role in MSC restriction by T cell. While in control mice, no activity was seen. There the T cell was killing the target cell from both F1 as well as B strain. Further, experiment evidence for the role of MSC molecules in T cell development was shown. Direct evidence for interaction of class 1 and class 2 MSC molecule displayed on thymic stromal cells with TCR of thymocytes during positive selection is provided by experimental studies with knockout mice. It was found out that class 1 knockout mice showed normal distribution of double negative, double positive and single positive CD4 cells, but lacked the population of single positive CD8 cell. Similarly, when thymic maturation was done in class 2 MSC knockout mice, it was found out that class 2 knockout MSC knockout mice had double negative, double positive and single positive CD8 cells, but lacked the population of single positive CD4 cells. It indicates that MSC molecules play very important role in development of single positive CD4 and CD8 T cells. Evidence for negative selection. To find out the role of importance of negative selection, here CD8 T cell clone was taken which has TCR, which is specific for HY peptide. HY peptide is a protein which is expressed in the male, the gene of which is present in Y chromosome. So it is a male specific protein. So a CD8 T cell clone is generated which has rearranged TCR, which is specific for HY peptide, presented on MSC molecule of B haplotype. So when a transgenic mice was generated with alpha beta rearranged gene which is specific for HY peptide on MSC molecule H2B. So transgenic mice, when they were generated and both male and female mice were produced, on analysis of thymic cells, thymocyte cells on male and female mice of the same haplotype H2B, it was found that both male and female mice shows normal present concentration of double positive thymocyte, single positive CD4 T cells, but female mice was showing the presence of single positive CD8 cells, but male mice lack CD8 positive cells, which indicates that HY peptide is a self peptide in male. So those cells which are specific for HY peptide, they were eliminated in male mice as compared to female mice, which indicates the presence, or which it serves as evidence for negative selection. Beside class 1 and class 2 molecules, which is very important for the selection process of maturing, for the maturation of thymocyte, peptides also has been found to be very important for T cell development. The role of peptides on class 1 and class 2 molecule during interaction with thymocytes are provided by TAP1 knockout mice. TAP1 is involved in transporting peptides from cytoplasm to endoplasmic reticulum, from where they get attached to cleft of class 1 MSC molecule. In TAP1 knockout mice studies, it was found that peptides could not bind to class 1 MSC molecule, and class 1 MSC molecules were not displayed on the surface. So, these mice, when their thymocytes were analyzed, it was found that these mice lacked the population of single positive CD8 cells. But when exogenous peptides were given to organ culture of these thymic organ culture of TAP1 knockout mice, it was found that class 1 MSC molecule appeared on the surface and CD8 cell population were found to be restored. Now, how this positive and negative selection happen? So, different hypotheses were given regarding positive and negative selection. The two important hypotheses were differential avidity hypothesis and differential signaling hypothesis. According to differential avidity hypothesis, it is a strength of interaction between TCR and MSC peptide complex in thymus which determine the distinct signal for selection. For example, no interaction leads to no signal, which result in death of thymocytes. Weak interaction leads to less signal required for survival of thymocytes, that is positive selection. Very strong interaction leads to more signal, which results in elimination of thymocyte or negative selection. So, differential avidity hypothesis believes that it is the strength of signal or interaction which determine the selection process. Next process is differential signaling hypothesis. According to differential signaling hypothesis, the interaction between TCR and MSC peptide complex involves different set of peptides which may lead to different signal. So, agonist peptide delivers very strong signal which leads to negative selection. Antagonist peptide deliver partial or weak signal which favors positive selection. In thymus, double positive cells, pro-T cells, undergo process of selection, positive and negative selection, and it generates single positive CD4 T cell or single positive CD8 T cells. Now again, hypotheses were given for the mechanism of formation of single positive cell. First hypothesis, instructional model hypothesis, states that interaction between peptide class 1 MSC molecule and double positive thymocyte instruct the cell to differentiate into single positive CD8 cell, whereas interaction between peptide class 2 MSC molecule and thymocyte instruct the cell to differentiate into single positive CD4 cells. Indicates instruction model hypothesis where whenever there is an interaction of double positive CD4 CD8 cell with thymic stromal cell representing class 1 MSC molecule, so this interaction instruct the cell to downregulate CD4 coreceptor and now this cell will generate into single positive CD8 cell. Similarly, interaction of double positive CD4 CD8 cell with thymic stromal cell expressing MSC2 molecule instruct the cell in such a way that it start showing lower expression of CD8 and high expression of CD4, and the cell further differentiate into single positive CD4 cells. Another hypothesis is stochastic or selection model hypothesis. It believes that there is a random downregulation of CD4 or CD co-receptor in double positive thymocyte. So, cell with high CD4 and low CD8 able to interact with class 2 MSC molecule and get survive. And if it doesn't interact, it will undergo apoptosis. And the cell with high CD8 and low CD4 able to interact with class 1 MSC molecule and get survive. If it doesn't interact with class 1 MSC molecules, it undergo death. Selection model hypothesis or stochastic model hypothesis, where double positive CD4 and CD8 cell randomly downregulate one of the co-receptor. So, if it downregulate CD4 co-receptor, and if cell with high CD8 and low CD4 molecule, if it interact with target cell with class 1 MSC molecule, it get survival signal and it survives into CD4 single positive CD8 cell, while the cell CD8 high cell with low CD4 co-receptor, if it interact with target cell with class 2 MSC molecule, it will get death signal and it will undergo death. Now, the double positive cell CD4 positive and CD8 cell with low or down regulation of CD8, random down regulation of CD8 co-receptor. Now, such cell with high CD4 and low CD8 molecule, if interact with target cell with class 2 MSC molecule, will get survival signal and they will survive into CD4 positive cell, while those cell with high CD4 co-receptor and low CD8, if get interact with target cell with class 1 MSC molecule, will not get survival signal and they will get eliminated by apoptosis process. So students, let us summarize what we have learned in this module. First, we have learned that T cell originate in bone marrow as T cell progenitor cells. T cell progenitor cells move to thymus where they interact with stromal cells and undergo proliferation and differentiation to form double positive pro-T cells. Large number of pro-T cells repertoire are generated with random combination of alpha and beta chain or gamma and delta chain. With enormous specificity for diverse number of antigens, the diverse alpha beta T cells repertoire undergo stringent round of selection where MSC peptide complex on thymic stromal cell interact with TCR receptor on thymocytes. The positive selection selects only those T cells which can recognize particular MSC molecule displayed on stromal cell, restricting their range of MSC recognition. The negative selection makes sure that only those MSC restricted T cells survive which cannot recognize self antigen. So, positive and negative selection make sure that T cell are self MSC restricted and self tolerant. These interaction led to generation of mature single positive class 2 MSC restricted CD4 positive cell which is also known as helper cell, and single positive class 1 MSC restricted CD8 cell, which is also known as cytotoxic TC cells. The mature single positive T cell then migrate to the blood and reach secondary lymphoid organs where they can interact with antigen. Thank you.